Hello, biology students. Today we're going to talk about transcription and translation. So far you've learned about DNA and RNA and how RNA is different than DNA. DNA, of course, contains all the genetic information, but it needs to be able to be copied. DNA likes to stay inside the nucleus, kind of like that one piece of paper that the teacher has that's their master copy of some assignment. They like to keep that one and make copies of it for distribution. And so many parts of DNA are copied little by little, and the way that they're copied is into something called messenger RNA. That messenger RNA, I'll show you here, is copied from the DNA inside the nucleus. Now you'll notice that the DNA inside the nucleus is bound by the nuclear envelope. And inside the nuclear envelope, there's these little pores called nuclear pores. The DNA is copied into what's called an RNA transcript, which is mRNA, messenger RNA. It leaves the nucleus through the nuclear pores and goes to the ribosome, here represented by this tan structure. The ribosome is responsible for protein production, as you know from the cells unit. But it's not that easy. Notice the interesting thing about DNA, as we learned before, is that A always goes with T. I'm having you look right here right now. A always goes with T, boom, boom. T always goes with A, and C always goes with G. That's the beauty of DNA. Because DNA is a template. One side of DNA exactly dictates what the other side of DNA would be. The one strand dictates the other strand. It's like having a key. A locksmith can actually make a lock based on a key. The same locksmith, though, could actually make the key based on the lock. If you have one strand of DNA, you know what the other strand's going to be. That's why DNA is able to copy itself so exactly every time. In fact, cells have to copy roughly 3 billion base pairs every time they replicate DNA. And that entire sequence of DNA is replicated with near perfection each time a new cell is made in your body. The way it does that is with this simple base pairing, A's with T's and G's with C's mechanism. Now, mRNA is an RNA, so it's single-stranded. You'll notice it here in the red. It is single-stranded instead of double-stranded, and it follows the same principle of production that the DNA does. What happens to make the mRNA is the DNA is uncoiled. Notice here, the one coil's up here, the one strand is up here, the other's down here, they're separated. And the transcription happens, and the transcription is the making of RNA, messenger RNA. The transcription process, the making of messenger RNA, functions on the same principle. You'll notice it makes a C go with G, another C go with G, and A go with T. There's one exception, and that exception is that the U goes with A. Well, what's U, you ask? Well, U is uracil. Do you remember that RNA has a few differences? It's single-stranded. It has ribose instead of deoxyribose as its sugar. And instead of having thymine, it has uracil. So normally with A, what would go with A? Oh, T would go with A. And the thing is, RNA doesn't have T. It doesn't have thymine. It has uracil. So when you're making an RNA transcript, an mRNA transcript, and there was a T in the DNA, there'd be an A in the mRNA, duh. And there was a G in the DNA, there'd be a C. If there was a G here, there'd be a C. All good. But if there's an A in the DNA, there'll be a U in the mRNA. Got it? Now here's what's beautiful. Every 3 mRNA nitrogenous bases, every 3 in a row, like U, C, C, form what we call a codon. So a codon is a set of 3 mRNA nitrogenous bases in a row. Here's why that's important. What happens is if you look right here, here's this red mRNA again. You'll notice here the codon is, if you can look real close, is U, U, U. What's interesting is there's another type of RNA that's very important, and it's called tRNA, if you follow my mouse cursor here, or transfer RNA. Transfer RNA has one job and one job only. It brings amino acids to the ribosome. If you remember, proteins are made up of amino acids. Ribosome's job is to make what? You got it, proteins. So, overall, DNA is transcripted within the nucleus into what's called messenger RNA. That messenger RNA carries a message, or it's called a transcript. That transcript goes to the ribosome, where it is quote-unquote read. But really what happens is the transfer RNA comes down and drops off one amino acid at a time. And you'll notice that on the transfer RNA there's a nifty little thing called an anti-codon. Well, you might think to yourself, why are they against codons? No, they're not against codons. They're just the matching thing to the codons of the mRNA. So if you look at the codons of mRNA, here, for instance, it's U, U, U. That's a codon. Or here, it's U, G, G. Notice they're in groups of 3. Or here, it's G, G, C. Groups of 3. But for this middle one, that's U, 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 the anticodon for tRNA that matches that is A, A, A. Because remember, U's are like T's. Okay, or over here where it was UGG, the anticodon that matches it is ACC. Got it? So what happens is there's this beautiful synergy. You've got DNA, two-stranded. They match each other. They unwind. One of those strands is copied or used as the base for making an mRNA transcript. That transcription process makes what's actually called a transcript, the mRNA piece. It comes out of the nuclear envelope, through the nuclear pores, down to the codon, where it is matched perfectly by a tRNA that has a corresponding anticodon. That tRNA has this very specific amino acid on it, and what ends up happening is you get an amino acid chain forming, and a chain of amino acids is called a polypeptide. The polypeptide forms what we know as a protein. The process of the polypeptide forming through the tRNA dropping off the amino acids is called translation. So transcription happens in the nucleus with the formation of the mRNA transcript from the DNA, and translation happens at the ribosome with the formation of the polypeptide from the amino acids. All of this based on codons. Now, here's how you do this. If you had a DNA strand that was AGT, what would the mRNA be? What goes with A? Well, T goes with A, Mr. DeWitt. Well, careful. mRNA does not have a T. Oh, uracil. Got it. Okay. U goes with A. What goes with G? C. What goes with T? A. Got it. Okay, how about this one? So CCA was the DNA template strand. What would the mRNA transcript's codon be to match CCA? G, G, T, right? Careful. mRNA doesn't have a T. Oh, yeah. It's U. All right. Now, what we learned already is that tRNA's anticodon has to match perfectly with mRNA. So what matches with a U? Well, an A does, Mr. DeWitt. How about with a C? G. And what matches with an A? So got to be careful. A U. So the tRNA anticodon here would be AGU. Now, some of you who are astute might notice that that looks a lot like the DNA, the kind of the parental DNA strand. AGT, AGU. What's different? Well, no T's in RNA, of course, so it's U for uracil. Okay, how about here? Well, what goes with G? C. What goes with G? C. What goes with U? A. Notice in this case, it's actually exactly like the parental DNA. So CCA is complemented by an mRNA codon of GGU, which is complemented by a tRNA anticodon of CCA. I think you get the idea. Now, the last thing is you need to be able to use what's called the amino acid codon chart. And there's lots of these available online, and your teacher will direct you to one. What you need to do is know that the codon charts are almost always based on the mRNA. So, however your codon chart works, you look at the mRNA codon that you have, and then you write the corresponding amino acid for that codon. The whole principle here, of course, is that each codon actually codes for a specific amino acid, which then will cause the polypeptide to have a specific ordering of amino acids. And with a specific ordering of amino acids, you get a very specific protein. So the whole concept here really is that this is a process of going from DNA to protein. And that's the power of DNA. That DNA actually, in its sequence of A's, T's, G's, and C's, 3 billion of them, actually commands certain proteins to be made, and even at certain times in our development. So that's all for today. This is the process of transcription and translation, how DNA actually turns into or codes for protein.